Hello, this is Alice Formiga of eOrganic, and I'd like to welcome you to today's webinar about managing insect-vectored viruses in organic tomato and squash. This is the second webinar in a series of 4 about biocontrol in organic vegetables. So today, I'm very pleased to welcome Dr. Rajagopalbabu Srinivasan, who's a professor of entomology at the University of Georgia. Dr. Srinivasan will discuss his research on a specialist predator for managing thrips and whitefly-transmitted viruses in tomato and squash. So now I will stop sharing my screen and Babu, you can share yours and unmute your mic.
All right, great. Good afternoon. Good morning, I guess, from where you are, Alice. Okay, so I'm going to basically be talking about viruses that infect tomato and squash, as Alice said, and I'm here at the University of Georgia, and this is what I mainly do, study insect-transmitted plant pathogens. And I'm stationed at Griffin. For those of you who do not know where Griffin is, it's about 45 minutes to the south of Atlanta. And this is a part of a OREI project that with the primary investigator being at Clemson University, and we are PIs who are actually in Georgia and collaborators on this project. So most of what I talk today will be about an introduction to insect transmission of plant viruses, and then I'll also talk a little bit about the project that we're doing and what the outcomes are and where we're willing to go with this down the road. My collaborator, two collaborators, the first one is Dr. Bemesia Tabassi, who's a plant pathologist and who's an associate professor also at the University of Georgia. He's located at Tifton, which is pretty close to the Florida state line. About 45 minutes down south, you'll reach the Florida state line. And we actually do a lot of our research down in Tifton, mainly because we have a very high vector pressure as well as virus pressure. And the next person is Dr. Tim Coulong, who's actually a professor of horticulture. Dr. Coulong is stationed at Athens, which is where our main campus is located. And Dr. Coulong is currently in charge of the organic program at the University of Georgia. All right, so what do I wish to accomplish in this webinar? As I said before, I'm going to basically give you an introduction to vectors. What is a vector and what are viruses that are transmitted by vectors, and how does the process of virus transmission take place? And then I'll follow up with a number of insect transmitted viruses that we regularly or routinely see here in the Southeast and in tomato and in squash. And then how does this information then translate to the ORE trials that we're currently doing? And I'll sort of wrap this up by giving you an idea of what other management options currently we're testing here and how those could be useful to manage these issues. Okay, so why are insect transmitted viruses a big issue? So I'm not sure if you can see my pointer, but if you could, I'm basically pointing to the pie chart on the left that shows, gives you an idea of the percentage of insect transmitted pathogens that are actually viruses. And you see about 2/3 of those are actually viruses, and insects are very efficient at transmitting these viruses when compared with other organisms that are also known to transmit viruses, mainly mites, fungi, and nematodes. So if you look at insects as such, there are quite a few of them. And on the next slide, I'll show you to which category they actually all belong to. But the most important point that I want to make here is that there are a number of vector species that are out there, but there are some what we call as super vectors. These are insects that are capable of transmitting numerous plant viruses. One example being Mysis persicea, or the green peach aphid, and the other being the whitefly or silver leaf whitefly, Bemesia Tabassi. So these insects are called as super vectors just because they are able to multiply really fast, and they're able to transmit a suite of plant viruses that are detrimental to numerous crops. And so here's a breakdown of the insect orders in which vectors have been described. These are the top three: Homoptera, Coleoptera, and Thysanoptera. And there are also a breakdown of number of vector species and the viruses that each one of these orders transmit. And you can clearly see Homoptera, these are the sucking, piercing and sucking mouthpart insects, namely your aphids and whiteflies, that can transmit a very high number of viruses. And I'll give you a brief outline as to why some of these insects are capable of serving as very good vectors. Okay. So we'll start with thrips. These are, there are a number of thrip species, over 7,000 thrip species that are currently recognized, but only very few of them are capable of transmitting plant viruses. This is because of very high evolutionary relationship between these two organisms, if you may call that. And then because of how they feed on a plant and the type of viruses they transmit. Most of what I'll be talking about today would pertain to tobacco thrips, or Franklinella fusca, which is a main vector of what we call as arthropods here in the Southeast, which includes tomato spotted wilt virus. And they have three different life stages, and all of them can transmit the virus, but in order for them adults to transmit the virus, they would have to acquire the virus at the larval stage. As I said before, this relationship is really complex, and they transmit by what's called as persistent propagative transmission, which means that once the virus is acquired, it is retained in the body of the insect. And propagative means the virus can actually multiply within the body of the insect, similar to many of the animal viruses or human viruses that we are used to. In fact, many members of this group have actually been, their ancestors are animal viruses, and still there are members in their family that can actually transmit viruses to animals. And if you're interested in looking at the life cycle, it's right here. As I said before, they acquire the virus when they're nymphs, but they can transmit it throughout their lifespan. And the pupae are actually inactive. And there are a number of reasons put forward to say why they can transmit the virus and why they cannot, but I wouldn't go into those details today in the interest of time. But the point that I'm trying to make is that these viruses multiply inside the gut of the insect. And what you see on the right-hand side at the top is basically in green is labeled virus particles that are present in thrips. So these viruses can multiply within thrips. And this is one example of the virus that they actually transmit. It's called as the tomato spotted wilt arthropods virus. This virus has got a very wide host range, can infect over 1,000 different host plants. But what you see here is a tomato plant that's showing classic wilt symptoms. And if you look at the leaves closely, you're going to see some ring spots. And the next virus, next vector, which is very important, is the Bemesia Tabassi, which is actually becoming important every year, more so because of the number of viruses that it transmits. And this virus is basically, we call it a cryptic species because there are over 40 different species, cryptic species within the same species. That makes them very deadly and makes them adapt to unique regions in the world and exploit the situation there or the systems there. And all this problem started to show up in the US in the 90s when we had this new biotype introduced from elsewhere. It's called MEM1, Middle East Asia Minor is what MEM stands for. And this particular biotype is very adapted exploiting plant species as hosts, and then they're also very adapted transmitting numerous viruses. And that's basically why we are interested in this particular virus, in this particular vector, and it's a clear definition of what you'd call a super vector. Anyways, I won't worry about it, but there are different groups that identify these with different taxonomic codes, but we'd call this as M1, and we'll just call this whiteflies for this seminar or webinar. So why are whiteflies such good vectors of plant viruses? And as you might have known by now, plant viruses are what you would call as obligate parasites, which means that they need living organisms and living cells for them to multiply. And this applies to pretty much all viruses. And what these whiteflies or aphids in general do is they're phloem feeders, which means they just feed on the phloem of the plants. And they actually have two types of saliva, and their mouth part is called as the stylet. And the stylet, through the intracellular regions, reaches the saliva. And once it reaches the saliva, they produce what's called as liquid saliva, and this basically allows them to acquire or dissolve plant nutrients and acquire the nutrients, and then they retract the stylet and then seal it up. It's called as gelling saliva, and in the process what they do is they actually keep the cells intact, and this allows the virus to multiply and on the plant, and this virus is introduced while they feed on the plant nutrients, and then when the stylet path is sealed, the cells remain intact, and thereby providing a perfect environment for the viruses to multiply. Okay, so this is why these insects, which use piercing and sucking mouth parts, are very good vectors of plant viruses. And whiteflies in particular transmit the viruses in two different modes. One is called a semi-persistent transmission, and the other one's called a persistent transmission. The difference between the two is the amount of time as well as where the virus is located. In the case of semi-persistent transmission, the virus is located in the foregut. It's called as foregut-borne virus, which means that the association between the virus and the insect is not prolonged, and it lasts for a few minutes to maybe a few hours. And the virus can be transmitted without a latent period. A latent period is when the virus is acquired, but the insect is not able to transmit it. So in the case of semi-persistent transmission, there's really not much of a latent period. Whereas in the case of a persistent transmission, which is in the case of whiteflies and even in the case of aphids and thrips, what happens is that, as the name indicates, the virus persists inside the body of the insect once it's acquired, and then it follows a circuitous route before it can be transmitted to the salivary gland, and from which the virus can be inoculated to a new plant. So what this means is that it takes quite a bit of time between the time of acquisition and being able to inoculate or transmit the virus, which is why there is a latent period. And these things are critical because this is what determines how you're going to manage the virus, whether you have opportunities to manage the virus based on the mode of transmission, and what sort of management options would you resort to. Okay. All right. So some of the examples, now that you've heard about what a virus is or what a vector is, how a virus is transmitted by a vector, here are some examples of viruses that are actual concerns of us in the Southeast and even on the West, in case this particular virus, tomato yellow leaf curl virus, is even in California and Texas. It's a DNA virus. If for those of you who don't know, it doesn't really matter. It's a virus, and most viruses are named after what symptoms they actually cause on a plant. So in this case, it causes yellow leaf curl on tomato plants. It's called as tomato yellow leaf curl virus, and this is basically how all the viruses are named as well. In case of HIV, human influenza virus, causes influenza in humans. It's called as human influenza virus. Same principle applies to how plant viruses are named. And you can see at the bottom here, the picture on the left at the bottom, it's a tomato field that's completely decimated by yellow leaf curl virus. And these plants are about 60 days old since they were planted. And you can see that they're all stunted, and every single plant in a field is infected. So really, and it shows you how serious the problem is. We can lose entire crops because of these viruses in the Southeast, and which increasingly seems to be the norm year after year. And And there's another virus. This is also found commonly in tomato. This is semi-persistently transmitted. The one that I showed you before is persistently transmitted. This is an RNA virus. It just gives you a diversity of things that we're dealing with here. And this is another recent one that popped up maybe not more than 10 years ago. It's the squash vein yellowing virus, also transmitted semi-persistently by whiteflies. And that's actually, it's a very serious problem in squash, but it's an even bigger problem in watermelon because it causes what you call as vine decline. And what you have there on the left-hand side is Dr. Scott Atkins and his technician Kerry looking at a dead watermelon vine caused by this particular virus. And it's watermelon is a huge crop in this area, and this is obviously a big problem, especially in Florida. Another one, cucurbit yellow stunting disorder virus, also semi-persistently transmitted virus, infects cucurbits. And this is another one, cucurbit leaf crumple virus. This is a DNA virus. It's also transmitted by whiteflies. Squash is susceptible to almost all of these viruses, and we have them all here at the same time. Every fall we try to grow squash. It's just sometimes turns into a nightmare because of these guys. And just to give you an idea of what a pathosystem would look like in terms of just whiteflies, gives you the number of viruses that we're dealing with in sort of interactions that we have with the whitefly and the other weed hosts. And what essentially starts as a problem becomes chronic, and because these things establish themselves in the pathosystem and there's almost impossible to get rid of. And I also want to tell you that this list actually is a couple of years old, and we've actually had to expand it because there's some other additions as well. So with the kind of diversity that I show you in this slide, one would expect that there are plants that can get infected with multiple viruses at the same time. So that's what this slide here shows. Mixed infection, double, triple infections are common. And oftentimes when you have these mixed infections occurring, it goes without saying that the yield losses and the symptoms severity in a plant increases tremendously, and the yield losses kind of follow that pattern as well. So what you have here on this slide, this is a plant that's actually not infected by the virus, which is represented by A, and what's represented by B is a plant that's infected with CYSDV, or cucumber yellow stunting disorder virus. And then what you have here on C is actually a plant that's infected by cucurbit leaf crumple virus. And what you have on D is a plant that's infected with both viruses. And you can clearly see the differences in symptom with these two viruses. And obviously, you're not going to harvest much squash from the plant that's infected with these two viruses. Okay. So that's the kind of issue that we're dealing with especially whiteflies. So that's, again, to give you a sort of an idea of the diversity of viruses that we currently deal with, and those that are actually transmitted by insects like whiteflies and thrips, and kind of reiterates why you call them as super vectors. Okay. So as far as this proposal is concerned, and this webinar is concerned, I'm going to restrict myself to just the trials that we do with the Organic Research and Extension Initiative, mainly with the trials that we employ a biological control agent. In this case, it would be the phytoseed mite Amblysia swiskii. So we did trials mainly in tomato and in squash. In our situation, the thrips trials are usually done in the spring, and squash trials are usually done in the fall, mainly being those are the primary seasons in which thrips and whiteflies occur. So if you are dealing with viruses, they're much more difficult to manage in almost in any situation, whether it's conventional or organic, just because a single insect can actually transmit the virus within a few minutes of feeding on a plant. So there are not many management options when it comes to that and stopping that. And also very low threshold. You're basically looking at no insect in a field. That's the threshold. If you see one, yeah, it's probably already too late. So these two things, particularly the very low threshold and the minimal management window that we have, makes it that much harder to manage plant viruses and control them, let alone doing this in organic settings. That just becomes that much more complicated. What you see here is actually a phytoseed mite feeding on a chili thrips or Scytothrips dorsalis. So this is a good biocontrol agent in terms of just managing the pest, be it either thrips or whiteflies, and that's one of the um, options that we chose actually for this particular grant, and it's to use this phytoseed mite and see how biological control influences pest reduction as well as virus reduction. So I'm going to run through the next few slides showing how the trials were done and what sort of results we got in each scenario. And then we'll follow that up with some interpretations later. So this is the trial that we did in 2019 spring, and some of this obviously has gone haywire because of COVID-19 restrictions, and follow that up with some bad weather. In 2020, we had a disastrous year, so some of the results that I'm going to talk about will pertain to 2019. So oftentimes biological control is good, but it's not what we call a silver bullet or a standalone option. So it would have to be combined with other options if you had any. In this case, we were lucky to have one, host resistance. That oftentimes is the best option to combine with biological control. And so in the case of tomato, and the virus that we are trying to manage here is the tomato spotted wilt virus. It's a thrips transmitted virus, and we used the predatory mite or Amblysia swirsky, basically obtained it from Cooper Biological Systems, and we attempted different application techniques. One would be to use the formulation that's mixed with bark, and that will be released at the base of the plant. And then we can also did another treatment where it was released on the foliage. And then they also had predatory mite sachets, and that was also used as a treatment. And then this was done with two different varieties, the Rosso Sicilian, which is the susceptible variety, and the other one is that SV7631TD is actually the resistant variety with the dominant gene governed resistance, namely SW5. So those are the different treatments and for the two varieties. And we did this in spring, and here are some photographs showing how the mite is being applied to the predatory mites. And we chose a curative rate that's suggested by Cooper and based on prior experiences, I believe. And we did 2 applications at planting and then 3 to 4 weeks after planting. And that's one of the collaborators actually applying the mites. So these are thrips numbers. Long story short, we found that the variety itself had some more effect on thrips, but the treatments did not. None of the treatments had any effect on the number of thrips that were found on these treatments, on these plants. So the application of mites by themselves did not vary with the treatment, or the application method did not vary with the treatment. And here's another one. We also then were able to monitor whether the mites were alive during this time and if they indeed differ based on the treatment and the application mode. Again, long story short, we did not find that the mites had any effects on thrips numbers as such. And we did find virus incidence. There was about 15% incidence in the case of the susceptible variety, Rosa Sicilian. We usually do not see any virus symptoms in resistant cultivars or genotypes, mainly because they produce what's called as a hypersensitive response, and then that pretty much kills the virus, and you don't see systemic infection following that. And at the top, you have pictures of tomatoes from a resistant genotype or cultivar on the left, and then you also see some symptoms on the right in the case of a susceptible cultivar. So not much of virus incidence as we would have liked, but we were able to see differences and then see if that translated into yield. So it did in the case of the resistant variety, SV763 1TD, as I said before, this is just an illustration to show these are large fruits that were harvested from tomato plants. And we did find that using resistance did increase yields, but application of mites with or without it did not make much of a difference in this case, as that's shown by the stats there. And then this is with unmarketable tomatoes. And here also we found that they were more in the case of the resistant variety. It's just that the resistant varieties were able to produce much more tomatoes or many more tomatoes when compared with the susceptible genotype. Okay, so that's with the tomato trial that we saw in 2019 in the spring season. We followed that up with the fall trial with whiteflies, because that's when we see a lot of whiteflies. That's the dry time of the year as well as the warmer time period. And our situation is also kind of unique in the sense that we grow over 1 million acres of cotton, and that sort of feeds into the system, and you get a lot of these whiteflies transitioning from cotton into cucurbits, and a lot of this was not really an issue until 10 years ago, but with the new introductions of viruses, the situation has dramatically changed. So this is a whitefly trial. We did this both with tomato as well as squash. In tomato, we try to manage the tomato yellow leaf curl virus. And then in squash, we used it for different viruses. I'll talk about that in the next few slides. But what you see here is that we were able to get tomato. Luckily for tomato, you have both resistance and susceptible. And Skyway actually is a resistant variety. And then the other one's the susceptible variety for tomato yellow leaf curl virus. Unlike the tomato spotted wilt virus, where you have hypersensitive response as resistance, here the plants get systemically infected, and the only difference being that they show reduced symptoms as opposed to hypersensitive response. And if the pressure is high, it sometimes gets hard to distinguish what's susceptible and what's resistant. So what I want to show here is that there are different treatments of mite application, same applying at the base, at the foliage, and with the sachet. And we did this for each variety, and we don't see much in terms of differences between variety here, but what I have on the left-hand side are the whitefly adult counts, and what I have on the right-hand side are the whitefly nymph counts. And you can clearly see within a single tomato leaf, these are leaf counts based on single leaf tomato plants from tomato plants on single leaves, and you can clearly see the number of whiteflies on each one of them. And application of mite has, I mean, if anything, a marginal effect. But this more pronounced in the case of nymphs. You can see some of the applications, especially in one variety application at the base, seems to reduce the number of nymphs, and mainly maybe due to the predatory nature of the mites that we're releasing. But these are counts of nymphs taken on a 2 square inch area. So you're talking about 60 to 70 on a 2 square inch area. So this kind of shows the pest pressure that we're dealing with is just, and you need 1 or 2 insects actually to feed on a plant for a few days to transmit the virus, or sometimes even a few minutes to transmit the virus. So the pest pressure that we have is just not conducive to manage these viruses. Well, do application of these predatory insects or biological control agents suppress pest population? The answer is yes. But is it sufficient to suppress virus incidence? And the answer then becomes no, for the most part. So here you can see every single plant that we had with the tomatoes that were planted, the insects were on them right after planting, and within a few weeks after planting, every single plant is infected. We didn't even bother to go ahead and get the yields at the point. But traditionally, we do see some yield differences, but in this instance, we did not, and it was also interrupted by an early frost. But traditionally, we do see that there are differences between susceptible and resistant genotypes, and that translates into yield. And that's what's shown here in this case. You see what you have on the left-hand side of this picture is that there is resistant genotype or cultivar, and then what you have on the right is a susceptible cultivar, and then you can see the differences in yield. And this is also often used in conjunction with other management options, namely so using mulch or different kinds of mulch or different kind of chemicals. But in this case, our chemical options were restricted mainly because of us using the biological control agent or the predatory mite. That's another area where we need to work more on to see how these mites could be compatible with other insecticides that are OMRI approved. And Oscar Liebert from University of Florida and Wagenbach at North Carolina are doing some research on that front right now. So using resistant sources or varieties when available would be the best way to prevent virus spread and decrease its severity and reduce yield losses. And other options could be useful at times and needs to be judged carefully when and how they can be applied. So switching gears, this is squash. We don't really have any resistance in terms of viruses in these varieties, and squash, especially yellow and zucchini, they're basically sitting ducks, and not much of virus resistance there, which is what we're working towards in with a number of other grants to breed for resistance. This is the same, different modes of mite application on the foliage and at the base of the plant, and using mite sachets. No resistant variety here, so just went with 4 treatments. And you can see application of mites does reduce the number of insects that you see on these plants, or this is per leaf count actually. And then you can see that the sachets actually performed a little bit better, but then you're also still looking at about 80 insects per leaf per week. So we're looking at an untenable situation here. That's just to give you an idea of what sort of whitefly pressure that you'd see on a single leaf. Okay, and then these are the immature counts, whitefly nymphs and whitefly eggs, and per leaf and a 2 square inch area. It's just tremendous amount of pressure that we're currently dealing with. And again, to answer your question, or say if application of these predatory insects or biocontrol agents, would they suppress pest populations? The answer is yes, and some more than the other. But would they be sufficient to reduce the amount of virus incidence in the field? That's just a totally different question. In this graph, you can clearly see that the numbers the mean separation letters show that there is reduced pest incidence or pest populations following application of predatory mites. These are 2 applications, one at planting and followed up by 3 or 4 weeks after planting. And we stopped releasing insects after that because by this time all the plants were actually infected with the virus. So here's again plants showing virus symptoms. This is plants. These are plants infected with cucurbit leaf crumple virus. And if you focus on the picture on the right, you can clearly see leaf crumpling and then yellowing, which is due to CYSDV. And you don't get any marketable yields with these kinds of pest pressure that we currently have. And again, we sort of repeated this in 2020, and we see very high numbers of white flies that are on these plants, and any management option basically at this point becomes untenable. Leave it, whether it's conventional or organic. So we definitely need to work more on this and identify options. But at this point, using biological control for this particular problem would actually not be of much benefit to the growers at this point. Well, going on, I just thought I could include some information that's based on studies conducted parallelly to the OREI trials. One thing that we were looking at is to look at different kinds of mulch. One will be the metallic mulch that you see in the middle, and the standard white mulch is what we would typically use in the fall and summer in the Southeast. And you can clearly see the difference between those 2 rows there. Metallic mulch has an ability to deter insects from landing, but again, it all depends on the kind of pressure that you're dealing with. If you have reduced pressure, these tactics are additive in the sense they build on each other and can help suppress pest levels and consequently virus levels. But you can see that some of these things can be helpful using a metallic mulch. And then what you have on the right here, where my pointer is, you do see what you call a live mulch with buckwheat, and that's helpful to an extent as well. So some of these options need to be considered along with biological control to sort of facilitate or suppress pest populations and reduce virus spread. And one of the other things that we've been looking at recently, and that has sort of increased our interest, is use row covers. And I think using enclosures like these can be really beneficial in organic settings as well as in conventional settings. Here's an example of how different using row closures or enclosures could be. These are hoops and with netting that was used until flowering. And you can clearly see that this is much better when compared to standard silver mulch, or even using the reflective mulch on the right hand side. So options need to be hedged and see which ones would serve better. You can keep the row covers for a longer period of time because that disrupts pollination and the amount of fruit set. And so everything has to be balanced for the crop to be profitable and even sustainable. So these are things that we're currently working on, and that could be sort of piggybacked with biological control and maybe something like induced systemic resistance, or trying to boost plants' own defense responses and things like that. That's all research that are currently ongoing, and these are techniques that could be applicable to organic production. But with biological control alone, we're not seeing all that benefits that one would expect. That's mainly because of the situation that we're having with the increased pest pressure. And so again, what would be the best option is to integrate a few options that we have, be it cultural or chemical, chemicals meaning that are sustainable and approved for organic production, obviously, and then exclusion, which is what I talked about in the last few slides with using netting, that could be beneficial in terms of keeping the insects away. And then obviously you need to factor in how long you can keep the netting in, and then uh, so as to not disrupt the pollination process as such, especially in crops like squash. And then factor in biological control with that could be beneficial. But there are instances, again, it goes back to the point that I made with the mode of transmission of viruses, biological control in certain instances, especially with non-persistent viruses, which I didn't really talk about today, which would be all the potyviruses that you typically see in squash with cucumber mosaic, zucchini yellow mosaic, and papaya ring spot or watermelon mosaic virus. All these are non-persistently transmitted, in biological control, and some of these instances have shown to increase virus incidence. So you got to be careful in choosing your management options, and you need to have a better understanding of the pathosystem and the knowledge on the pathosystem to make decisions. And some of our future trials would concentrate on integrating some of these options, basically to reduce virus incidence. And there are numerous options that are available for suppressing pest populations that shouldn't be an issue if you're just trying to manage pests. But the issue is when they are able to transmit plant viruses, and with even a few insects in a field can do that, but with increased pest pressure, that becomes that much more challenging. So that's basically in a nutshell of what I had. So I'll stop there and acknowledge some of our funding sources over the years, from either USDA related grants and OREI, as well as from SARE, that went into doing some of this work, and our lab group there doing all the hard work, basically. So I'll just stop there and see if we can entertain some questions, and if you have any comments, I'll be happy to discuss those as well. So at this point, Alice, do you want me to go ahead and stop sharing it?
No, you can keep sharing the screen if you like. Yeah, you can just leave it up there.
Okay.
Yeah, but I just, yeah, that was an interesting webinar, and it's too bad and you haven't found the magic bullet yet, and it
Yeah.
really is unbelievable. But it is how much pest pressure you have, but in a way, it's very useful to know that certain things like biological control on its own might not be effective. So um, we're going to start our question and answer time. So if you have a question, feel free to type it into the Q&A box on your screen. We'll just be reading them out loud for the next half hour or so until we run out of time. And if you don't see the Q&A box, again, there should be a black bar with some controls at the bottom of your screen. And if you click on the Q&A one, that should pull it up.
I
Um, yeah,
Yeah. Now
okay,
I see one from Dawn.
okay.
How does one dispose of infected plants? That's a good question. There are, like, if you're in the Northwest, when I used to be in Idaho, we would what we call as roguing, remove infected plants, potato plants, bag them in and discard them. But here we just have too many plants that are infected, so we just plow them in. So that's basically what we do here.
Okay, I got some other questions that had been coming in before as well. One person wanted to know whether your tomatoes and cucurbits are being transplanted or grown directly from seed in your field.
Well, yeah, that's a good question. The ones that we used are transplanted. Some and seeding oftentimes does not work well for us, because the moment these seedlings sprout from the ground, you see insects on them. So it's in that situation, it works better to transplant them, and especially tomatoes, they work much better being transplanted.
Okay, and then also the second question there was, your population level of whitefly is amazing. How does the whole field get infected?
That's been our billion-dollar question since the last 5 years. And like I said, the population situation that we have is sort of unique because we do have a lot of cotton that we grow, and it's a time when, but in the fall, early fall, they start defoliating cotton, and all these white flies sort of migrate to vegetables, and they love squash plants. Um, so that's a problem that's basically made by us, I guess. But in the past, I mean, not even 5 years ago, this was not an issue because we didn't have this much of virus issues. We didn't have these new introductions of the virus. So yeah, like I said, it's usually not that difficult to manage whitefly populations, but when it comes to managing virus, that's where it becomes very difficult.
Yeah. Okay. We had a question also about zucchini yellow mosaic virus. Someone's been using Bovaria bassiana on the seed to protect against the disease and was wondering what other people are using.
Well, that's like I said, zucchini yellow mosaic virus is a non-persistent virus, and it can be transmitted really fast. It's similar to your potato virus Y if you're in the Northwest. So really not much in terms of management for organic production. Stylet oils have been shown to be effective, but you guys know what the issues with using stylet oils are. Number one, it's expensive. Rain washes it away. We have rains here almost every day in the summer and fall. So reapplication is another issue. But they do offer some sort of protection. Another way to maybe do something would be to increase induced plant resistance or systemic resistance in plants. We haven't done much work on that, but the group in Auburn with John Murphy, they did quite a bit of work, and it seems to have some effect. I don't really know what commercial products are available to deal with it at this point, to be honest.
Okay. Um, are there any catch crops that could be used for the whitefly or trap crops?
That's a very good question. Oftentimes in tomatoes, we use squash as a trap crop because that's squash is a whitefly magnet. Unfortunately, when you're trying to manage that in tomatoes in squash, then it becomes difficult. There are some that could repel whiteflies like marigold and things like that, but there are a lot of ways, like you said, to manage whitefly populations. But even there are organic options to manage whitefly populations. But the issue is with the virus.
Okay. Um, do other controls that inhibit feeding, like stylet oil, have an effect on virus transmission?
I see that question from Mike. They do to an extent. Like I said, the issue with stylet oils is that you have to frequently apply them, and there could be phytotoxic effects as well. I haven't done that with squash, but we've tried with potatoes for quite some time, and there is some degree of success. Then the issue is you just continuously have to depends on where you live and in the Northwest, and especially in some parts of Idaho, it doesn't rain as much and stylet oils work. But out here where we are, it rains every day. So that's another issue.
Okay. Do you know if calcium may work blocking pathogen movement? Have you tried that?
I have not, and I can't comment on that because that's not something that we've tried. Maybe worth looking into, but we haven't tried it.
Have you been practicing crop rotation with tomatoes and cucurbits, or are the same crops grown in that plot every year?
We do transition between row crops sometimes, but honestly, with tomatoes and even beans, they're all rotated. So yeah, the same crops don't grow on the same field every year. Um, but the point is, these are not big farms. We have, growers that grow them side by side all the time. So yeah.
Okay. Um, have you done any work with other biocontrol organisms such as Delphastus or Incarcia for whitefly or Cucumeris for thrips? And then a comment, Swirsky is okay in certain thrips applications and minimally useful for whitefly.
Yeah, we haven't done much work with other biocontrol agents. That's something that we're planning to do. Um, the other person, I don't know if Oscar Liebert has given us webinar, but he's actually he's someone who's actually worked with Delphastus for quite some time, and he may be able to comment more. Um, but they are all options, and we have even thought about using some entomopathogens in the near future, but we haven't done that yet. Those could be options as well. Yeah.
Okay. Are there any known viruses that we can spray to control the western flower thrips? We do see some good efficacy from repeated applications of essential oils, but it costs a lot of money to make multiple applications.
Certainly. So I'm guessing the question was about entomopathogenic virus, some kind of like a baculovirus or something like that. Not that I'm aware of. I haven't seen or heard of any virus that's capable of infecting thrips and suppressing pest populations. So I did say that the virus that we are interested in, the tomato spotted wilt virus, does multiply in thrips, but it does not kill the organism. And they go about doing their business. They may live a few days longer, but it doesn't really kill them. So that's, yeah, we don't have a pest control virus as we have in the case of lepidopteran insects like a baculovirus or a granulosis virus. We don't have any of those available for thrips.
Okay. Are you doing anything to support predatory insect populations that would help control the whitefly? The comment is I didn't see any flowering hedgerows or interplanting of plants that would support predatory insect in the photos.
Sure. Well, I didn't show that particular trial. Well, we were been planting buckwheat, and then we've planted some crucifers in the past for thrips in the case of tomatoes, and uh, they do help to a certain degree, and they help build predatory thrips in the case of tomatoes, and number of other predators in the case of squash. Yeah, but it does reduce their numbers a little bit. And in fact, if you recall, I did show a slide with live mulch on either side of the plastic rows. It helps to a degree. Yes, it certainly helps to a degree, but is it enough to suppress viruses? That's a question. That's a debate.
Okay. Um, is transmission in the greenhouse, even before setting in the field, an issue? And if so, is there a rapid diagnostic test to determine whether the transplants are already infected?
That's a very good question, and in fact, in tomatoes, that is a big issue. And they don't show symptoms in the greenhouse, and when you put them in the field, they would show symptoms within a week. That's a serious problem for us, especially when we have these open air production greenhouses, as we call them. It is a problem, but what we're working on that as well. We are trying to use a number of these sort of systemic resistance or India systemic resistance enhancers in plants in the greenhouse, along with netting. And that seems to reduce what you actually bring into the field in terms of inoculum. Yeah, that's a good point, and that's one of the things that we're working on, and the goal is to minimize what you actually put into the field as an inoculum source at planting. Yeah.
Okay, so we have someone who grew Georgia candy roaster squash, and after the squash was harvested, the flowers continued to set and were filled with the spotted and striped, probably the cucumber beetle, and he would pick the flowers holding many beetles and crush them, and he plans to grow and harvest beetles early in the season.
Okay. Yeah, that's in fact, those cucumber beetles also can transmit what's called as, I think it's called bacterial wilt caused by Erwinia. That's an issue as well. I wouldn't want to really grow a lot of those beetles. Um, yeah.
Yeah. Okay, and any observations on predatory effects of phytosized mites or whitefly nymphs or adults, including thrips on tomato?
We did see quite a bit. I mean, we didn't see any predatory, observed predatory activity as such, but what we did see, what we did do was monitor uh, uh, um, predatory mite numbers as when we sample either whiteflies or thrips. We also sample for phytoseiulus. We do see them, and so they establish themselves after we introduce them into the field, but we didn't directly observe any uh, feeding behavior. We did see some in the microscope, but yeah.
Okay, well, let's see. I don't see any more questions in the queue, but we still have a few minutes, so I'll just give another couple seconds in case anybody wants to type in some more questions, and there's a good likelihood that we'll have time to answer them. And I just want to let you all know as well that I'll be sending everyone who's on the webinar a copy of the slides after the webinar as a PDF along with an evaluation survey. And we'd be really grateful if you could fill that out. And the recording, as I mentioned before, will be available within the next 1 to 2 weeks, along with the last one in our series on the eOrganic YouTube channel. And we have another couple of webinars in this biological control series, which will cover different aspects of biological control coming up, including Dr. Oscar Lilberg, whom Babu mentioned, and then we have another one. So that one is on the 24th, and then we have another one coming up on March 1st. So you're very welcome to register for those and attend them. So since I don't see any more questions, I'd like to thank you so much, Babu, for presenting this webinar, and thank everyone who submitted questions for all the great questions. And thanks everybody for joining us today.
Thanks, Alice. I appreciate you guys attending the seminar, and maybe next time I'll have better answers.
Okay, thank you. Bye-bye.